Hello and welcome to this presentation on human papillomavirus and its ability to cause cancer. My name is Luke Farmery and I am a trainee histopathologist working at the Royal Devon and Exeter Hospital. A lot of what we do as histopathologists involves looking at tissue under a microscope, which is called histology, or by assessing individual cells, which is called cytology. The images here show koilocytes, which are cells infected with HPV. On the left is when they're individual, and you can compare it with the background cells on the right-hand side of the image. And then the right-hand image is when the koilocytes are situated in the tissue they came from. But most of the time, histopathologists are interested in the diagnosis of cancer. So why pay these cells any attention? Well, some of you may be already aware that in addition to the traditional effects we think of when considering infectious diseases, there is also a strong association between chronic infection and cancer. HPV itself is known to cause several types of cancer listed here, including cervical cancer, which we'll go on to talk about. Cervical cancer is a very common cancer involving the lower part of the womb or the cervix. HPV is actually found in 90% of the cancers involving the outer part of the cervix, which is where the vast majority of the cancers in this region occur. So, if cervical cancers are caused by a virus, and we can vaccinate against the virus, then surely we can prevent the cancer. And indeed, this is true to a degree. With the introduction of Gardasil, a vaccine for HPV, that was introduced in 2006, with the intent to do just that. But this only protects against certain types of high-risk HPV. And these two phrases I'll go on to explain after we look at HPV a little bit more closely. So HPV is a small DNA virus which is transmitted from human to human by intimate skin-to-skin contact. The genome is circular and comprises roughly 8,000 base pairs which are arranged into 8 genes. 2 of the genes are of particular importance in oncogenesis. These are E6 and E7, both of which suppress tumor suppressor genes. Coming back to types then. There are hundreds of types of HPV, but the 2 main types that contribute to the risk of developing cervical cancer are HPV 16 and 18. These are therefore deemed high risk for the disease. Different types are distinguished by differing from each other by over 10% in a highly conserved L1 region on the genome. Each type of HPV has many variants to it. 2 of HPV 16's variants are highlighted here, the European group of variants and the non-European group of variants. The literature already shows that having a non-European variant HPV has a 10-fold risk of developing cancer, despite the fact that this variant differs from other variants of HPV by only around 200 nucleotides. And it's examples such as this that have inspired the use of next-generation sequencing to further explore the genetic variation in HPV and try to associate any variations with the carcinogenicity of the virus. So, this brings us on nicely to the paper that I have selected, Deep Sequencing of HPV 16 Genomes, and this paper set out to assess the genomic variability and associate any such variation with the ability to cause cancer. They started with 55,000 cervical samples, a bank that had been acquired from a local screening program. They then took all of the specifically positive for HPV 16 samples in women of greater than 21 years of age and in those individuals who had not opted out of having their cytology used for research purposes. The end number for this, after these exclusion criteria were applied, was 796, 69 of which had cancerous samples, 472 had high-grade but precancerous changes, and 255 had low-grade or no lesions within them and were deemed as controls. It was anticipated that the viral load between samples would be different and would potentially confound the results unless accounted for. Therefore, samples were stratified into low and high viral load depending on the signal strength produced by a type-specific dot-blot oligonucleotide hybridization technique. The next step of the methodology involved the use of custom degenerate primers to amplify the HPV 16 genome. The resulting amplicons were sequenced and assembled according to a reference genome. Because this study highlighted a novel approach, there was a strong emphasis on quality control and concordance with the gold standard, which was considered by this paper to be Sanger sequencing. The method used actually had an excellent completion rate of 98%, with internal concordance on repeated samples of 99.7% and concordance with Sanger in the samples that were compared of 99.97%. Only 2 nucleotide positions dropped below 85% concordance, as demonstrated in the graph shown here, and this was in a region that had proved difficult to amplify. The figures shown here are heat maps which demonstrate the sequence depth. Samples are on the y-axis, amplicons are on the x-axis, corresponding to areas on the HPV genome. Cells highlighted in green indicate a good 100 times depth. Red indicates less than 2 times depth. The sequence depth was high in 80% of samples. A further 6.9% of samples had high quality data but with specific amplicon dropout. 3.3% of samples had large central low sequence depth but later was interpreted as genomic deletions rather than amplicon dropout. And then a further 9.6% of samples performed poorly across most amplicons. And poorly performing samples are strongly associated with low viral load, as shown by these violin plots. The median percentage of amplicons exceeding 25% times coverage can be seen to be greater than 90, whereas this drops to 85% in low viral load. This table details the 961 SNPs detected in this study. The group compared their results to 62 previously sequenced HPV genomes in a paper that used Sanger sequencing and found that of their 961 SNPs, 642 were new or novel. 55% of these novel SNPs were actually code changing. Changes in CpG sites were also documented. The paper goes on to state that CpG methylation of the HPV genome is highly associated with pre-cancer as opposed to benign infection. It's hypothesized that the loss or gain of these sites could be one genetic factor contributing to carcinogenicity. In this paper, the HPV-16 European lineage had the greatest number of CpG site losses and site gains, with the most frequent gains being in the E2 region. But further research is needed to assess the relevance of these findings. 719 of the samples were eligible for determining the variant of HPV they contained. 604 samples contained European lineage variant, whereas 115 had a non-European lineage variant. Samples containing the non-European variant lineage were shown to have a strong association with cancerous samples, with an odds ratio of 4.3. This is supported by the background literature that suggests non-European variants are a higher risk for cancer. Due to the depth of sequencing data, this study was also able to tell when one sample was co-infected with 2 HPV-16 variants, and this was demonstrated using the IGV software, and could demonstrate consistent variations for 2 variant lineages at specific nucleotide positions. As mentioned, 26 samples were shown to have very low reads in the central genome, often involving the E2 gene. These samples also had a strong association with cancer, with an odds ratio of 27.3. This could potentially be linked to E2's function in suppressing E6 and E7, and could also be a marker of HPV host integration. In summary, this paper highlighted a high throughput method of sequencing HPV-16 with high quality data concordant with the gold standard. This method had additional benefits in detecting novel SNPs, CpG changes, and the ability to detect co-infections of HPV-16 variants. Confirmation of the non-European variant HPV-16's association with cancer was possible. The technique also detected central genomic deletions in a subset of samples, a finding strongly associated with cancer. However, the study had a low sample size, which limits its statistical power. Furthermore, 9.6% of the samples were of low quality due to low viral load, hindering analysis. This study also makes no mention of controlling for the lifestyle factors or comorbidities of women, despite mentioning access to electronic health records and evidence in the literature that these factors, especially smoking status, affect risk. Lastly, the relationship of types of other high-risk HPV infection was ignored, although perhaps this was outside the remit of this particular study. So in the future, There could be work to look at the circulation of different HPV-16 variants and determine whether they interact or influence each other in developing carcinogenicity. We could also look to assess whether the central deletions are truly associated with high-grade lesions. Or there could be more work on co-infection with other types of high-risk HPV. And indeed, thinking more broadly, other cancer types could be researched, potentially with extraction of DNA from paraffin-embedded tissues. Thank you very much for listening, and here are some references which are also included on the handout.